Hi everyone, I'm Jess Bailey and I'm an embryologist and chief cryobiologist at Boston IVF. And I'm joined today by Marsha Forman, who is the senior embryologist and lab manager for our New York sites. And we were recently talking, hi Marsha, we were recently talking and we realized that we have a lot of patients who are really asking us quite frequently about our embryo grading. And we thought that maybe we would take the opportunity to have a little discussion between us and talk about what might be helpful for our patients to know and guide them through their upcoming cycles about what to expect in relation to their embryo grading, because there's so much that happens during an IVF treatment, and we really want to make sure that they understand what we're talking about when we're discussing the quality of their embryos. So I think this is a great starting point for patients coming up with cycles.
Yeah. Well, let's get right to it then, okay?
Sounds perfect.
Time for your biology lesson, everybody. So when discussing embryo grading, I think it's really important to remember that the grading refers to the appearance of an embryo at a certain point in time. So most often we're grading embryos at the day 3 and then the advanced stage of day 5, 6, and 7. So when grading embryos at the day 3 or cleavage stage, we use two variables. One is the rate of growth, which is measured in how many cells are present at the time when we're doing the observation. And the other is a degree of fragmentation, or bits that haven't really divided out appropriately. And we measure those using letters. So Jess, if you were to kind of describe fragmentation to your patients, how would you describe it?
So fragmentations are an interesting thing because I think when we first start talking about it, it sounds very scary to a patient to be like, there's fragmentation present. Yeah. And it doesn't need to be scary in any way. It's just one of those other features that we can visually see under the microscope that give us a little bit of guidance for grading. And just like any other cellular organism, every cell has some sort of metabolism that's happening. And as that metabolism happens, we have some waste that happens. So a lot of times that fragmentation is the metabolic waste from the embryo that it's going to spit out as it's dividing. And we also have mechanisms for self-correction. So as the embryo divides and we get more cells, those chromosomes are overlapping and they're doing all that biological work that they should be doing. But in the process, if they find any errors, they will self-correct and they will spit that stuff out so that they're not carrying it on as the embryo develops. So when we're looking at it, we're seeing how much of that activity is occurring. And we do that on an ABC scale. So, or D, hopefully we're not giving too many Ds. And that's based on the percentage of those fragmentations that we see present. So A would be that it's exceeding our expectations. We only see maybe about 5% fragmentation. It's very clean. It's not having a whole lot of work needing to be done, and it's not spitting a whole lot out. B's are pretty similar to A's. We take it up to 20% fragmentation, but really no concern at that point. That's still a normal level. It's still meeting our expectations of what we'd like to see for fragmentation. Once we get to C, that's kind of more of in our still developing stage. It might be having a little bit more fragmentation than we'd like. It kind of piques our interest to say, is it overcorrecting itself? Meaning, is there a lot that it needs to fix? Because no matter what happens, the embryo wants to be perfect. It's going to try to correct anything that it sees in the process that maybe might need some tweaking. So once we get to C, we have up to 50% fragmentation. Beyond 50% fragmentation, that's a big red flag for us, and those are Ds. And generally, we don't give out too many Ds. We don't have those kind of embryos in general, but those are embryos that we kind of see peter off, and they don't usually develop too much more than that. Most of the time, we are dealing with A, B, and C embryos. And like I said, C is more still developing. It doesn't mean it's out of the game. We have plenty of babies that come from C grade day 3 embryos.
Right. I think that's an awesome description. I usually do use like a food analogy when I'm talking to people, and I tell them, like, if you break like a loaf of bread, like your two cells that are dividing and a couple crumbs fall out, it's okay. Your bread is still good.
Yeah, exactly.
If you, like, break open a loaf of bread and it like falls into dust in your hand, like that's not so good. So we don't want to see that.
Right. It might, yeah, if it's falling apart as you eat it, that's not a good sign.
It's not good bread. Okay, so let's talk about, as the embryos continue to grow and develop, not every embryo is going to make it to that next stage where we do grading, and that's going to be your blastocyst stage. So a blastocyst is like a fluid-filled ball of cells with another little tiny ball inside of it. And the outer aspect of the blastocysts are what we call the trophectoderm, and they will make the aspects of the pregnancy that hold the pregnancy in place, like your placenta. And the little tiny ball that's on the inside of this fluid-filled cavity is the cells that will make the baby, or we label those as the inner cell mass. Obviously, both of those components are going to be very important to making and sustaining a good pregnancy. So Jess, can you talk a little bit about how we grade these? Because it is different. This is where I think a lot of the confusion comes in with our patients, because there's not this uniform way of grading embryos on day 3 versus day 5, 6, and 7. So how do we grade embryos or blastocysts at this stage?
Right. And I think that's an important distinction, because on day 3 embryos, all the cells are essentially the same. They all have the same components, and they have no definitive destiny yet. Whereas when we get to blastocysts, we have those two different cell types that are forming. They're differentiating into those cells that will become the placenta versus cells that will become the baby part. And so we do grade them separately. Before we grade those, we always look at the expansion of the embryo. And I think the expansion of the embryo can be a little overwhelming, again, when we're talking to patients, because we're talking about giving a number about how far the embryo has started to expand and really define those two different cell types. It's important to remember that every embryo is going to go through all of them. It's not a big deal if on day 5 we take a snapshot observation of this embryo and it's a grade 3, which I'll explain more in a moment, versus an embryo that we look at on the same day that's a grade 5. That one that's a grade 3 is still going to develop and make it through a 5. It's just they all are going to grow at their own pace, just like each of us in our lives as we're developing, all developed at a different pace. But here we are as adults, and we can all do essentially the same basic things. It's the same thing with your embryo. So there's no concern to be like, oh my God, I only had a 3, but my friend had a 5. It's just a snapshot at that moment. If any of those became a baby, they went through all of them, because you have to be.
They can expand and contract. I've seen that. I check one in the morning.
Frequently.
It's big. It's like a size 5. And then I go back to freeze it, and it's like a 3. It's a little.
Yes, they're constantly changing. So the way we go through that entire gradation of expansion, we start at what we call a morula, where you're just seeing a mushy mess of cells. You don't have any of that differentiation yet. And then we have 2 early embryo stages where you're starting to get a cavity. The embryo is starting to show signs of differentiating into those cells, but we don't see full differentiation yet. And that fluid-filled sac that happens inside the embryo, depending on how much of that is present before full differentiation, those are 1s and 2s. So those still would be great embryos. They still have great potential. They just aren't gradable in the same way as it is once we get to the 3 grade. Once we get to the 3 grade, we have a full expansion of that fluid. And you can really see the difference between what are the embryos that become the part of the cell that becomes the ICM, the inner cell mass, versus the part that will become the trophectoderm. 4 is once we have full expansion. And I love that you have this picture behind you, Marsha, of the embryo development, because I'm going to talk about the zona pellucida real quick, which is the shell around the egg. And you can see that right there. That's perfect. That's a nice 4-cell day 2 embryo with a zona pellucida.
In like a weather person.
Yeah, that worked out great. And that would be a 6 over there. But as that differentiation happens and that fluid-filled sac expands, that portion, that shell, starts to thin out. And when it's completely thinned out, that's what designates your embryo as now a 4. So the difference between the 3 and the 4 is just the zona. They both have full expansion. They both have that big fluid-filled sac, and you can still see the full ICM and a full trophectoderm for both of them, but the thin zona differentiates them.
Right.
Then as they develop a little bit more, we get to a 5, where that zona pellucida is kind of cracked open, and the embryo itself is starting to come out of the shell.
This is the part that completely blows everybody's mind, because our embryos hatch out just like chickens hatch out of an egg, even though it's not exactly like that. But they do. They kind of ooze out, and so they leave that shell behind, and then you have this beautiful, like amoeba almost like structure of the trophectoderm, and you can see the little tiny ball of cells in there that's going to be the fetus.
Yes, exactly. So once it comes completely out, it's not inside the zona. That look like 2 separate things in the media. That's when we have the 6. The zona pellucida, that eggshell, gets left behind. It'll get reabsorbed and expelled out of your body if you were having a natural pregnancy. And the embryo itself, at that grade 6, uses the trophectoderm cells to implant into the uterus. and support the growth of the inner cell mass, which will ultimately become your baby. So that's the grading system as a whole. So again, it's not a concern if you are transferring a 3 versus a 5. It can continue that expansion in the body. I mean, that's what happens naturally.
Yeah, so Jess, let's talk about that. So how do you grade that trophectoderm, and how do we grade the inner cell mass? Because we grade them differently.
Right.
So
when we're looking at them, they're obviously different cells, right? So with the inner cell mass, we're looking for a nice compaction of a lot of cells. And the trophectoderm, it's kind of like a cobblestone of cells that kind of protect around the baby. And we kind of grade them very similar to the way we did fragmentation, right? We're looking at an ABC scale based on the number of the cells that are available. And that A scale is the A grade is the best, meaning that it's exceeding our expectations. We have tons of cells there. B,
and they're
it's still great.
Yeah, they're communicating.
So for inner cell mass, we have lots of cells, and it's fully packed down. It looks like a nice little pack of cookie dough on the side of the embryo. Like it's nicely packed in there. It's been rolled up. It's going to make a great chocolate chip cookie. It's there. The trophectoderm, same way. You want lots of cells, and it's like cell. We see a nice little bubbling cell. It's like bubble wrap right around that.
Yeah, totally.
Yes. Then when we get to the B grade for each of these, it's still a good amount of cells. It's meeting our expectations. It's not like, oh, my friend had an A and I have a B. A B is what we want. A B is great, and that's why we freeze them. And we'll talk a little bit more about that later. But a B means it's meeting our expectations. We don't have any concerns. It has a great potential to continue on. And the same thing for both ICM and inner cell mass. The primary difference with the inner cell mass is that we might not see as much compaction. We still have a lot of those cells, but it might still be like a little bit more of a fluffy dough. Like you still need to pack it down more. Then we get to the C. Now C means still developing. It doesn't mean that it's falling apart and dead. It just means it's still developing. So it needs more time to grow, more time to proliferate, and get us more of those cells to be able to pack it down for the inner cell mass, or create a more concrete bubble wrap that can implant into the uterus and protect that growing embryo.
Yeah. I think that's an excellent way to describe it. I usually tell people, I've never heard the bubble wrap, but that's nice because they're so uniform and perfect. I usually tell people like the trophectoderm on the outside kind of looks like a quilt. We want to see all the patches are perfectly put together. I mean, you may have another quilt that has one huge patch and a couple tiny little patches. It doesn't mean it's a bad quilt. It just means like that one patch, that one cell is really stretched thin. And so do we like to see that? We don't really like to see that, but it doesn't mean that it's not a viable embryo.
Right. So a lot of times if you're getting a C grade on day 5, it may be that a third of that embryo has a lot of those flat areas where there's like giant cells. It's not really completely patched together in a uniform way. But if you let it grow another day, because we're going to look at it again on day 6, those embryos can still become B's and A's. It's not impossible for a C grade trophectoderm to become an A grade for trophectoderm in 24 hours. We've seen it. And it's kind of nice to let them grow if they need to grow, not to rush them, because you can also see information the other way. So if it doesn't continue to grow and you start to see cells falling apart, that's an indication that one embryo is not your best choice. And we should be looking at these other embryos that are really developing and giving us a higher potential to implant and give you your family that you're looking for.
I think you hit the nail on the head there, Jess, because I think one part of us going through and doing IVF in the laboratory is the goal to try to get somebody pregnant. The other part is scientifically looking at what's happening with this patient and help them have an understanding as to why they're not getting pregnant. And then the other part of that is, the longer we can hold them in culture, the more selection occurs. So we know that if we transfer a day 3 embryo, the selection process for implantation is probably like 33%. But if we hold them out to day 5, where not every embryo that was growing on day 3 makes it to day 5, the selection process is already starting to happen. So we have a much higher implantation rate when we transfer a day 5 embryo over a day 3 embryo. And it's not because one is better than the other. It's just that they've selected themselves out now. So it makes our job
I mean, we see that naturally every month when we're trying to get pregnant too, right? Just because you didn't have a positive pregnancy test in that month doesn't mean you didn't have an embryo that fertilized, tried to go through stages. It could be that it was developing, coming down the fallopian tube, doing exactly what it should do, and then just stopped growing. And that is, like we were talking about day 3 versus day 5, there are inherent things that happen in the embryo that are critical at day 3. And so the control of the genetic components of that changed at that point from what was being controlled by the egg itself and really the mother from that egg. It makes some switches in its controls, and now the embryo itself is in charge. And so if that embryo is not going to continue to grow, it's going to stop right there. And if we've already selected that embryo for transfer, you're not going to know, did you not implant because it stopped growing, or are there other factors? So sometimes it's a benefit if you have a lot of embryos to choose from to allow them to grow and get us more information and have a better selection process and increase your chances for that positive outcome.
Right. I want to take this chance to remind people, because I find, you know, we started this conversation out talking about embryo grading, but also about what kind of questions do people ask us. One thing I find our patients are often confused about is that the day you have your egg retrieval is actually considered day 0. So I think patients leave after their egg retrieval and think that's day 1, and they start counting out and they expect, you know, to hear from somebody on day 5 or day 6 or day 7. But if you're already starting one day off, you're expecting information much earlier and we're ready to give it to you. And that can be super confusing. So just remember, the day that you come in for your egg retrieval is actually day 0. Day 1 is the day of fertilization, and you count it out from there. So, Jess, what other questions do you find you're most often asked with your, you know, when people are asking you, you know, what's going on in the lab?
Well, I do a lot of the cryo stuff. So I think a lot of people ask me about what is a good quality embryo? Like, why don't we freeze everything? And I think it's important to remember that embryos, when we're assessing them for transfer back into the body, is a little bit different than we're assessing them for biopsy and freezing, because we have to consider the events that are happening in freezing or biopsy and freezing in the embryo. When we're transferring an embryo, we're just taking that observation of the embryo that we saw and putting it into the body, someplace that it's very comfortable with and it knows what it should do. So if you have a still developing C-grade trophectoderm, it still has a great chance of implanting. It's going to continue to grow and do exactly as it would if it had come down the fallopian tube and landed in the uterus.
Right.
Once we start adding in treatments, if we take a C-grade trophectoderm and we freeze it, there's less cells available to recover from those events. The protocols we have are fantastic, and the survival rate in general is 96%, but those are based on good quality embryos, which are A's and B's. So when we add in the C's, we do get great survival. That is true. We have it's lower. I don't know the exact number, but it is lower. But the positive outcomes for the pregnancy rate is definitely lower. And it may be that transition that even if it survives a thaw, it's not reinitiating the growth it needs, because it has so much more growth it needs to catch up after these events.
Right.
And like when we biopsy.
That trophectoderm is the embryo's first line of defense against the cryopreservation process. You know, we do the best we can with the techniques that we have. And like you said, Jess, our stats are excellent. And, you know, there's always evidence and research going on to make sure we're doing what's best for our patients. You know, but that trophectoderm has to survive the cryopreservation and the thawing process in order to be, you know, viable on the other side. So
well, the trophectoderm implanting into the uterus is that first step. Regardless of how nice your inner cell mass is, if you don't have any trophectoderm to dig into the uterus and plant its roots so that it can protect the embryo as it grows, you can't develop the pregnancy. You need the placenta to grow the baby in. So that's the other part when we're doing our biopsy. If we're going to biopsy an embryo before we freeze it, we can't really do that to a grade C embryo, because that means too few cells, right? It hasn't met the expectation. It's still developing.
You want to explain really quick, Jess, what is biopsy? Because there may be some people who are watching this for the first time that don't understand what biopsy is.
Absolutely. So some patients come through and want to have some genetic testing on the embryo so they can make sure that it has a normal complement of 46 chromosomes. And they can check for aneuploidy and make sure you have 2 of each of the chromosomes you're supposed to have, and then 1 XY or 2 X's, depending on the gender. There are also patients who come through who may have a family history of a genetic disorder like cystic fibrosis or Huntington's disease, and they would like to make sure they don't pass that disease on to their child. And so this is an additional testing that we can do in the embryo, where we would remove some of the cells from the trophectoderm, send them off to the genetics lab to assess for the genetics, and freeze the remaining embryo while we wait for those results.
Right.
When we get the results back, anything that came back as unaffected for the disease we checked for, or have a normal euploid, meaning it has the 46 chromosomes that we were expecting, are suitable for transfer. And we can use just those embryos to transfer back in a subsequent thaw cycle, where we can thaw that embryo and put it back into the uterus. Now, when we do that process of removing some of those cells, we have to make sure we're leaving enough cells behind to implant. Because if we take some cells off and they come back euploid, that doesn't do us any good if there's not enough left when you put it back in the body to do everything it needs to do to produce the outcome that we're looking for. So that's again where the grading happens. So sometimes on day 5, you have a biopsy embryo and it has some cells available that we can take, but we're like, you know what, if I take this, we're going to be kind of close to whether or not we feel like we're going to have enough remaining. Let's let it grow another day and see if it gives us some more to work with. And a lot of times it does. And we can wait, get some more cells, get a B or an A trophectoderm going, then take some cells off for the genetics lab, freeze, and then we have the outcome that we're looking for.
Right, right. That's an excellent description. So good. This was really fun today.
I know. I hope this is helping people because, a lot of times we're in the lab and we don't always talk face to face with patients, but I know you and I, Marsha, have a lot of patients who call and ask questions. And so it's kind of nice to be able to give a perspective from the lab. I also know sometimes they're calling nurses who have all the things that we've said to them, but to kind of get the answer from us who are looking at them every day, I think can be reassuring. So I appreciate that we were
snapshot of being able to watch the embryo from soup to nuts, from the day the eggs were retrieved, the injections, and all of the notes that we have in the embryology laboratory. Sometimes we can give a different take on it than even, our nurses or our physicians have. So this was really fun. And maybe people will have questions they can type in and we can do it again.
Yeah, absolutely. If anybody watching has questions that you want to hear more from the lab, anything from Marsha and I or anybody from our team, please put it in the comments for the video and we will take a look at that, and hopefully we can address more of your questions and make your journey through Boston IVF a little bit more understanding and make it less stressful and more relaxing so that you kind of know more what's going on.
Yeah, that was awesome. Thanks, Jess.
Thank you.